Thank you very much. So, good afternoon, everybody. If you're in the UK, obviously, if you're in America, it's going to be a different time of day. So, it is my absolute pleasure to introduce the first of our DRIIMB online seminars. So, DRIIMB is a collaboration between CCP Biosim, CCPN for NMR, CCP4 for X-ray crystallography, and CCP, what have I missed out? EM for Biosim, N for NMR. So, of course, what we're interested in, you can gather from the collection of people in the collaboration, is integrative molecular biology. So, how we integrate all of these different experimental tools together and combine them with simulation. So, it is my amazing pleasure to welcome Brinda from the PDB. So Brinda is at Rutgers and she is an expert in integrative biology, and she is associated with the integrative biology structures in the PDB. So I'm going to hand over to you now, Brinda. We're very, very much looking forward to your presentation. Thank you very much.
Thank you, Sarah, and thank you to the DRIIMB team for organizing this special seminar. Let me share my screen. All right, is my screen visible?
Yep, it's good.
Yep. All right, okay. Thank you, Sarah, for the introduction. I'm Brinda Vallat. I'm a member of the RCSB Protein Data Bank, and I lead the PDB-IHM project at Rutgers University. Today, I'll be presenting our work on developing a new system called PDB-IHM for archiving integrative structures of macromolecules. Let me begin with a quick introduction to the Protein Data Bank. As you all may already know, PDB is the single global repository for experimentally determined structures of biological macromolecules and their complexes. It was established in 1971 with 7 structures and currently contains over 338,000 structures of macromolecules. It is the first open access digital resource in all of biological sciences. PDB promotes the FAIR guiding principles for scientific data management and makes structural biology data findable, accessible, interoperable, and reusable. The PDB archive is managed by the Worldwide Protein Data Bank organization, consisting of the RCSB PDB in the US, Protein Data Bank in Europe, Protein Data Bank Japan, the Biological Magnetic Resonance Data Bank, BMRB, and the Electron Microscopy Data Bank, EMDB. The PDB archives structures of macromolecules determined primarily using X-ray crystallography, NMR spectroscopy, and 3-dimensional electron microscopy. The structures archived in the PDB contain the 3-dimensional atomic coordinates and associated metadata. The PDB also archives certain structures that are determined using multiple methods. These methods usually use one of the traditional structure determination methods, such as X-ray crystallography or NMR or EM, in combination with other methods such as neutron diffraction or small angle scattering. Recently, structures of many macromolecular assemblies have been determined using integrative modeling methods. Integrative structure determination is an innovative approach to modeling the structures of biological systems based on data produced by multiple experimental and theoretical methods. It is typically used for modeling structures of complex macromolecular assemblies that are not amenable to traditional methods of structure determination. Integrative modeling involves combining data from multiple experimental methods, such as electron microscopy, NMR spectroscopy, small angle scattering, chemical cross-linking, etc. The experimental data are gathered and converted to spatial restraints and combined with known starting structures of molecular components using computational modeling algorithms to build the structures of complex macromolecular assemblies. So then the question is, can the PDB archive integrative structures? There are some technical challenges to archiving integrative structures in the core PDB archive. These challenges arise due to some of these factors. Integrative modeling involves restraints from multiple experiments, such as chemical cross-linking, mass spectrometry, small angle scattering, etc., that are not supported by the PDB. Integrative models can be multi-scale. They're not necessarily atomic and can consist of both atomic and coarse-grained representations. These models can also contain compositionally heterogeneous assemblies and models belonging to different conformational or compositional states. Integrative modeling can use starting structural models that come from experimental or computational methods, and they can use complex modeling methodologies and different software applications. In order to address these challenges involved in archiving integrative structures, the Worldwide Protein Data Bank set up a task force consisting of researchers from different fields contributing to integrative structural biology. The first meeting of the task force took place in October 2014 at EBI in Cambridge. The task force came up with a set of recommendations for addressing the challenges in archiving integrative structures. They recommended that structural models, data, metadata, and workflows need to be archived. A flexible model representation needs to be adopted. Methods need to be developed for assessing model uncertainty and model quality. A federated network of archives need to be created for structural models and experimental data, and publication standards need to be established for integrative modeling studies. In this talk, I will present how we've addressed some of these recommendations over the past few years. Our goal is to build a system for archiving integrative structures based on the recommendations from the task force. So first, what are the requirements for archiving integrative structures? The first requirement is the creation of data standards. Data standards are the primary requirement for archiving, and these enable automated data processing as well as dissemination of data in a standard form. Data standards encompass definitions for the data that need to be archived, and in our case, this includes chemistry of macromolecules, small molecules, and definitions for the 3D model coordinates. They also need to support information required for model validation, such as the spatial restraints and starting models used as input data. And they also need to include descriptions for metadata information, such as authors, software, samples, experiments, workflow, etc., that help in interpreting these structures. The second requirement is to create software tools that support the data standards. And finally, we need the archiving system that helps with data collection, processing, archiving, and distribution. Together, this constitutes the data pipeline for the archiving system. So let's look at the first requirement, which is the creation of data standards. PDB-XMM-CIF is the data standard used by the PDB to archive structures of experimental structures of macromolecules. PDB-XMM-CIF specifies the standards for representing macromolecular structures, including the chemistry of polymeric macromolecules and small molecules, atomic model coordinates, reference sequence information about the sequences and small molecules, and additional metadata. PDB-XMM-CIF overcomes the limitations posed by the legacy PDB format, which lacks explicit data definitions. It requires fixed field and column positions for data items and cannot handle large structures. PDB-XMM-CIF is designed to be extensible in order to handle evolving methods in structural biology, and has extensive software support for reading, writing, and checking the compliance of the MM-CIF data files. We decided to extend the PDB-XMM-CIF data standard for integrative modeling. This extension is called IHM-CIF, where IHM stands for Integrative and Hybrid Methods. We retain the standard definitions for representing polymeric macromolecules and atomic model coordinates. These are retained from the main PDB-XMM-CIF data dictionary. and new definitions required for integrative modeling are created in the IHM-CIF extension dictionary. The extension dictionary includes descriptions of multi-scale models, multi-state and ordered models, and conformational ensembles. I'll go into these descriptions in a couple of slides. The extension also supports definitions for compositionally heterogeneous assemblies, the various kinds of experimental restraints and starting models used in integrative modeling studies, and also information regarding experimental data and other information that could be residing in external resources. We also developed a software library called Python IHM that supports the IHM-CIF data dictionary. This is developed and maintained by Ben Webb in Andre Charlie's group at UCSF. The library provides a mechanism to programmatically generate MMCIF files compliant with the IHM-CIF dictionary. Both the IHM-CIF dictionary and the Python IHM toolkit are distributed open source via GitHub. Next, we created the infrastructure for data deposition, processing, archiving, and distribution of integrated structures. Deposition involves submission of model coordinates, spatial restraints, starting models, and additional metadata. Data processing involves curation, model validation, visualization, issue of accession codes, and releasing the structures. The data distribution system includes a website that provides search and browse functionalities, access to downloading the model coordinates and validation reports, and tools for visualization. Together, these constitute the data pipeline for the PDB-IHM archiving system. The PDB-IHM data pipeline is underpinned by the IHM-CIF data standard, and the infrastructure for deposition and processing system has been built in collaboration with Dr. Carl Kesselman's team at University of Southern California using their DRIIMB scientific asset management platform. Access to PDB-IHM is available through two related websites. One is primarily for depositors and curators and handles the data harvesting, deposition, curation, and validation. And the other website is for data distribution, where users can freely search, find, and download all relevant information for released integrative structures in PDB-IHM. Let's now look at the different components of the PDB-IHM data pipeline, starting with data deposition. The deposition system and the underlying IHM-CIF dictionary provide support for archiving multi-scale, multi-state, ordered models, and ensembles. What do we mean by this? Ensembles are a set of models that correspond to multiple solutions that individually satisfy the input restraints within a certain threshold. Multi-scale refers to models that have both atomic and coarse-grained representations. Multi-state models correspond to models in multiple conformational or compositional states that collectively satisfy the input information. For example, there can be structures of macromolecules in bound and unbound states or open and closed states, and they collectively satisfy the input information. Ordered states are multi-state models that belong to a time-ordered pathway or another ordered path, such as an enzymatic reaction or a biochemical pathway. In addition, we can have any combination of these four features represented in an integrative model. As recommended by the wwPDB-IHM task force, we have adopted a flexible model representation that can handle multi-scale, multi-state, ordered models, and conformational ensembles, as well as models with heterogeneous composition. Another important deposition requirement is submission of starting structure models and spatial restraints from different experiments used as input in the integrative modeling investigation. These are essential for model validation. Experimental and computational models of components in an assembly that are used as starting models can be included in the deposition, and these definitions are supported in the IHM-CIF dictionary. Additionally, specifications for describing spatial restraints derived from experimental data such as chemical cross-linking, electron microscopy, small angle scattering, etc., all these can also be provided. to enable capture of a broad range of generic distance restraints, such as those from, say, mutagenesis or coevolution data, a general representation of distance restraints between features at different granularities, such as between individual atoms or residue ranges, are also supported. In addition, we also want to capture the provenance information regarding the starting models and the spatial restraints, which can be provided either by referencing the source information from external databases, or they can be referenced via digital object identifiers. A generic set of definitions are provided to handle the steps involved in the modeling workflow. These steps can include model representation, configurational sampling, and analyzing and validating the resulting integrative models. Providing the modeling protocol information during deposition makes it easier to interpret the structure for any downstream applications. The deposition system supports harvesting and collection of heterogeneous data and metadata. The requirements for deposition are IHM CIF compliant files that include all the necessary information, including the model coordinates, input spatial restraints and starting models, external references to sequences, UniProt sequence information, and other chemical moieties, and related metadata. Users can either start with fully compliant files to breeze through the deposition process, or can build a compliant file starting from a very minimalistic MMCIF file using the data harvesting section of the deposition workflow. The data harvesting workflow consists of systematic steps that guide users to start from uploading model coordinates, and then enter additional metadata manually, and then upload starting models and spatial restraint files. The final step is the creation of a compliant MMCIF file that can be submitted for curation and further processing. As mentioned earlier, if the depositor starts with the fully compliant file, then most of these steps can be skipped. Additionally, the PDB-IHM system provides an API for bulk deposition and curation of a collection of structures belonging to a single investigation or publication. Multiple simultaneous depositions can be carried out using the deposition API. After deposition, the curation process begins. PDB-IHM data curation involves both manual and automated steps to ensure data consistency and completeness, compliance with the PDB-X MMCIF and IHM CIF data definitions, and checking the reference sequence and small molecule information and standardizing this information, visualizing models using either Chimera X or Molstar visualization software. We then issue accession codes to the deposited structures and also generate validation reports. Methods for validating integrated structures have been implemented based on recommendations and guidelines provided by the wwPDB-IHM task force. The current version of the report includes assessment of model composition and model quality assessments for all integrative models, and data quality assessment and fit of the model to the experimental data for structures based on small angle scattering and cross-linking mass spectrometry. These are currently supported. Validation of structures based on other kinds of data, such as 3D EM and FRET, are currently under development. The validation software has been integrated with the deposition and curation pipeline so that preliminary validation reports can be obtained after deposition, and the depositors can use this information for the manuscript review process. Summary and full validation reports, PDF reports, are available. And we also have a mechanism to generate interactive online validation reports in HTML format. And these are available on the PDB-IHM website for all release structures. Development of validation methods has been a collaborative effort involving experts in various experimental methods and members from other data resources. For example, we worked with the SASBDB repository and members of the small angle scattering community to develop validation for SAS-based integrative structures. And we worked with the PRIDE data resource at EBI and members of the structural proteomics community to develop validation methods for cross-linking MS-based integrative structures. The collaboration with PRIDE involved extending their MZ-IDENT-ML data standard. They use the MZ-IDENT-ML data standard for mass spec data. So we had to collaborate with them, work with them to extend that to incorporate new definitions, new cross-link specific definitions, as well as creating a separate section within PRIDE called PRIDE cross-linking to handle deposition of cross-linking data. We've also developed mechanisms to automatically pull relevant data from both SASBDB and PRIDE to support the generation of validation reports for SAS-based and cross-link-based integrated structures in PDB-IHM. Here I show selected plots from sample validation reports specific to SAS and cross-linking MS, and all these were generated in collaboration in extensive discussion with experts in the community. There are two software applications that enable visualization of integrative structures. The Chimera X desktop application and the Molstar web application both support visualization of integrative structures. There is a Molstar extension toolkit called MolView Spec, which also supports data-driven visualization and can be used to visualize both atomic and coarse-grained integrative structures. Chimera X also supports visualization of associated data such as 2D EM images, 3D EM maps, cross-linking restraints, and the starting models used. They can be superposed with the final models to understand how the starting models were used in the modeling. The image shown here corresponds to the integrative structure of a 552 subunit nuclear pore complex from yeast, archived in PDB-IHM and visualized using Chimera X. The model was obtained using spatial restraints from 3D EM cross-linking and small angle scattering data. This slide shows a snapshot of selected structures archived in PDB-IHM. The structures vary from small and medium-sized complexes such as the COP9 signalosome and BB-some, which are atomic, to large complexes such as nuclear pore complex, which is multi-scale. Here I highlight a few recent examples from PDB-IHM. This figure depicts the assembly mechanism of the nuclear pore complex elucidated by integrative modeling and has led to the creation of spatiotemporal models of the post-mitotic intermediates involved in the assembly pathway. The modeling was carried out using data from 3D EM, cross-linking, SAS, and I think it was fluorescence correlation spectroscopy. The study reveals that the assembly pathway of the complex follows two distinct molecular mechanisms where the order of incorporation of the structural components is reversed. And this information can be obtained from the deposited structure in PDB-IHM. New integrative modeling methods are also being developed to understand structural dynamics of macromolecules. For example, shown here is the integrative structure of a large GTPase, human guanylate binding protein 1, determined using restraints from fluorescence resonance energy transfer, small angle scattering, and electron paramagnetic resonance spectroscopy data. The study illustrates different conformers involved in the oligomerization and reveals important mechanistic and kinetic information regarding the conformational transition between multiple states. So this is a multi-state model. Following the success of machine learning-based methods such as AlphaFold and Rosetta Fold in modeling protein structures, expansion of deep learning-based modeling algorithms to include experimental restraints is an interesting approach in integrative modeling. Shown here is an example from AlphaLink, which is a new method that incorporates experimentally determined chemical cross-linking restraints into the network architecture of AlphaFold to improve performance and predict distinct.
And Brinda, the slides are not advancing. The last slide we have here is the structures in PDB-IHM. Okay, now this is the big one. Then we have this one. We don't see the next one.
I'm still on this one.
Okay, all right, okay.
Okay, so I mean, this was the nuclear pore complex, the multiple states of the GTPase, and this is the AlphaLink structure. So yeah.
Okay, sorry.
That's okay. So the plot here on the right shows the rate of growth of integrative structures over the past few years, and the corresponding plots for X-ray, NMR, and EM structures in the PDB. The overall trend for integrative structures is promising, and the scope for applying integrative approaches to model large macromolecular assemblies has further expanded because of the massive pool of starting models provided by deep learning-based methods like AlphaFold. This slide shows the statistics for different types of experimental methods used in determining structures archived in PDB-IHM. The rows represent the different experimental methods, and the counts correspond to the total number of entries that use a given source of input information. The columns represent particular combinations of the input information. These are the counts of the total number of times these combinations occur in entries in PDB-IHM, and these black circles indicate which methods are included in these specific combinations. As we can see here, cross-linking mass spectrometry is a predominant method used in integrative modeling, either alone or in combination with other methods, followed by 3D EM, NMR, SASFRET, and hydrogen deuterium exchange. A 2023 analysis of the primary publications corresponding to low-resolution 3D EM structures deposited in the PDB showed that approximately 25% of such structures are likely to be integrative structures where other types of experimental restraints are used along with the low-resolution 3D EM map. And many of these structures use cross-linking MS data, but are not annotated appropriately, leading to data loss. PDB-IHM provides a robust mechanism for archiving such structures with complete set of information regarding the input restraints and starting models, as well as modeling methods used in such integrative modeling studies. I'm just showing an example here, which is based on 3D EM and chemical cross-linking and archived in PDB-IHM. Depositing such structures to PDB-IHM not only makes the data fair, but it also enables enhanced structure validation based on the full complement of experimental data used in modeling. PDB-IHM was developed separately from the PDB to enable an agile development platform and to develop the infrastructure required for archiving integrative structures. However, from the very beginning, our goal was to create this infrastructure and then unify it with the PDB so that the capabilities of the PDB can be expanded to archive integrative structures. And in the past year, we achieved that goal. In 2024, we unified PDB-IHM with the PDB archive, which further enabled integration with the RCSB.org website for data dissemination. RCSB.org now supports search, access, and analysis, as well as visualization of integrative structures. The integration with PDB and RCSB.org was primarily made possible because of the common shared data standards and IHM CIF being an extension of PDB-XMM CIF. Shown here is a snapshot of the structure summary page of an integrative structure on the RCSB.org website. It's very similar to what you will find for experimental structures. You can see the image. You can visualize using Molstar. There's other metadata about the primary publication, authors, and also IHM-specific information. There are two distinct aspects of the PDB-IHM unification with the PDB archive, as well as integration with the RCSB.org website. This figure shows the current PDB-IHM data pipeline, including newly added extensions that support integration with the PDB archive. Integrative structures are now archived alongside experimental structures in the PDB archive. The repository contents include the MMSIF files, validation reports, and additional holdings files similar to what you can find in the core PDB archive. The PDB-IHM directory is mirrored by the wwPDB partners. Most importantly, we now issue PDB accession codes for new structures, as well as already released structures where have also been issued PDB accession codes. We also have a synchronized weekly update process where integrative structures are updated and released along with experimental PDB structures at 0 UTC on Wednesdays. Along with the weekly update process, new PDB-IHM entries receive digital object identifiers, which allow online versions of primary publications to be cross-linked to the entries in the PDB archive via these digital object identifiers. Simultaneously, both the PDB-IHM website and the RCSB.org website are also updated with new entries that are released or updated weekly. The second component of the integration corresponds to updates in the RCSB.org website. Integrative structures are now fully part of the RCSB.org website, alongside experimental structures from the core PDB archive and selected computed structure models that are also available on RCSB.org. These computed structure models are from the AlphaFold database and model archive. All RCSB.org APIs and search services now support integrative structures, the search API, data API, and other sequence coordinate API services. All these APIs support integrated structures. Individual structure summary pages are available, as I showed earlier, and integrative structures can be visualized using the Molstar software available on the website. All the added annotations from external resources, such as Go annotations and UniProt annotations, are also available for integrative structures. IHM-specific information, such as input experimental data and starting models and references to data in other resources, such as PRIDE and SASBDB, these are also available on the Experiment tab of the integrative structure page. So what are our next steps in the short term and the long term? We encourage the structural biology community to deposit integrative structures to PDB-IHM. We plan to work with scientific journals to promote structure deposition. We also want to reach out specifically to the 3D-EM community to encourage deposition of 3D-EM-based integrative structures to PDB-IHM. Through the PDB-IHM pipeline, it finally goes into the PDB archive, but through the PDB-IHM pipeline to prevent any data loss. We will continue to expand the system to support evolving integrative modeling methods. We will also continue to develop validation methods and incorporate support for validating models based on other kinds of experimental data, in addition to SAS, cross-linking MS, and 3D-EM. We will work on improving the interoperation with other data resources to streamline access to pre-release data for structure validation. We're also working on developing Bayesian methods for structure validation applicable across different structure determination methods. Following one of the initial recommendations from the wwPDB-IHM task force, one of our long-term goals is to create a federation of interoperating data resources that contribute to integrative structural biology. This goal poses additional challenges such as development of common data standards and tools for federated data exchange. In 2019, a workshop titled Working Towards Federating Structure Models and Data was held during the annual Biophysical Society meeting at Baltimore in Maryland. The workshop focused on addressing challenges involved in creating a federation of integrative structural biology resources. The outcomes of the workshop have been published in a white paper. The figure shows a conceptual representation of the data exchange in the proposed federation. At the center here are the structural model resources, including PDB with PDB-IHM and the Model Archive, which is the repository for computed structure models. The outer circle depicts different kinds of experimental data used in integrative modeling, such as 3D EM, SAS, cross-linking, NMR, etc. The arrows represent data exchange among the different structure model and experimental communities and the corresponding data resources that support integrative structure determination. Common data standards currently exist for some of the data types used in integrative structural biology. Extensions of PDB-XMM CIF have been created to support small angle scattering data archived in SASBDB. It's called SAS-CIF. There's also the NMR exchange format, the NEF format, and the IHM CIF, which is the extension for integrative models. We also have an extension for computer structure models called Model CIF, which is used by Model Archive and AlphaFold DB. And we also have an extension of PDB-XMM CIF for fluorescence and FRED data called FLR CIF. Since the 2019 Biophysical Society meeting, collaborative efforts have emerged across different scientific communities to promote open science and fair data practices. Here I highlight the outcomes from some of the community activities focused on creating data standards, benchmarks, publication guidelines, and validation methods, specifically in the small angle scattering community, the cross-linking MS community, FRET, hydrogen deuterium exchange, and EPR communities. As part of the PDB-IHM system development, we've collaborated extensively with the cross-linking community and the PRIDE repository for extending their data standards, archiving cross-linking data, and developing mechanisms for efficient data exchange. To summarize how far we've come, when the PDB was started, we had static structures of single molecules, such as the structure of myoglobin determined by X-ray crystallography at atomic resolution in 1958. Since then, the scope of structural biology has expanded, allowing for larger and more complex structures to be determined using different experimental methods. Today, we are already archiving floppy coarse-grained structures of macromolecular assemblies, such as the yeast nuclear pore complex, determined by an integrative approach, archived in PDB-IHM, and this is in the nanometer resolution scale. In the future, we think that structural biology will involve tackling newer and more complex structure determination challenges, such as elucidation of the three-dimensional structures of complete genomes and creation of a spatiotemporal and dynamic model of cells. I'd like to thank all the RCSB PDB team members for supporting the PDB-IHM project. Specifically, I would like to thank our director, Stephen Bowley, our director emerita, Helen Berman, who has joined us today, and our UCSF PI, Andre Shali, for their support and encouragement. Special thanks to my colleague and mentor, John Westbrook. I'd also like to acknowledge Arthur and Jared from UCSF, who've been working on the IHM validation software package and developing Bayesian validation methods, respectively. And also Monica, who's the PDB-IHM biocurator here at Rutgers. I want to thank other Shali Lab members, specifically Ben Webb for developing and maintaining the PDB-IHM software, as well as our collaborators at University of Southern California from Carl Kesselman's team, especially Hung Suda, who's been helping us with building the PDB-IHM deposition and curation system. I want to acknowledge the support from all the wwPDB directors and members of the IHM task force and the ModelSIF working group for their recommendations and continued support. I want to thank the developers of Chimera X, Molstar, as well as the FLR-SIF team at Dusseldorf, and all the researchers who have deposited structures to PDB and PDB-IHM. And last but not the least, I'd like to thank the United States National Science Foundation for funding the development of PDB-IHM, and the RCSB PDB core operations are jointly funded by NSF, NIH, and the Department of Energy, U.S. Department of Energy. Thank you all for your attention. Please visit pdbihm.org and rcsbpdb.org to deposit or download structures and learn more about integrative structures archived in the PDB. And I'm happy to answer any questions.